All right. Two problems has to be solved to make alternative proteins are commercially available. Human beings can consume. The first one, the problem is allergenicity. Second one is anti-nutritional factor. Allergenicity, you must be knowing that, right? For example, some people say I'm allergic to milk, right? I have a friend, if he or she, I should not name the name person also because we are on camera, and that person eats crab or shellfish, whole skin becomes red and there's a boiling here, right? Allergic, people are very allergic. We're talking about Maggie packet, and as I said, if you took and look into the ingredient, some things they write in the bold, be careful. Nowadays, if you go to restaurant, they clearly ask you, if you're allergic, please let us know before you. They don't tell it in the small restaurants. They don't care about you. No, they do care about you, but that's another story. If you go to hi-fi restaurant, they ask you, are you allergic to some kind of ingredients? You say, oh, I'm allergic to nuts. Then they don't use nuts in the food, right? Because allergenicity is one of the biggest challenges that we are facing today. Because sometimes they're less severe. If you have a small bubble on your lower teeth, it will go in half an hour for me. It's not a big, I don't care, to be frank. But some people get breathing problems, and that can be life-threatening. Am I clear? That can be life-threatening. So allergenicity has to be taken care in a very careful way. In some cases, it's a less problem. In some cases, it's really a severe problem. Okay, there are 8 foods in America they classify as big 8 allergenic food: wheat, fish, milk, eggs, peanuts, soybean, tree nuts, and crustaceans like crabs and shellfish. They are allergic, and the restaurant should ask before if there is any seafood product and something like that. They label it carefully. And I think in the Americas they also have a rule, they have to mention which one is allergenic in the packaging. I don't know if there's in India. It's also in India now. Okay. FSI has an. When I was I'm on Indian, of course, it was not there at that time. They were not very strict about that, but now things are very strict. Okay. Now, what I want to tell you here is that alternative proteins can also be allergic. Can you see here soy? Where is soy? See, soy can be. See, soybean is allergic, but alternative proteins we're getting ingredients out of it. But still, we need to be careful whether they are allergic. Am I clear? Why I want to tell you, but I'll come back to that one a little bit later. I don't teach you about how allergic as happens. There's a lot of big figure, but when you're free, go and read all these things. But I'll tell you in a very simple word, in 1 minute, how allergy happens. Okay? Forget about this big figure. Let me try to simplify it. There is something called allergens, okay, and something called body, right? We eat food as food, not as allergens. Okay, we don't know whether it's allergen or not. We just eat it as a food. When it comes to stomach, there is a digestion or bioavailable or many things will happen. There's a lot of absorption. Many thing happens in your body, right? There is something called antibody in our body, right? Antibody. Okay, and they are very specific to antigens, right? You studied in immunology lectures, right? When this antibody sees some molecules as they are maybe a food particle, imagine protein is actually good one, but sometimes body thinks it has an antigen. It has an okay. Then they interact with each other. Okay. Then the immune will trigger in our body. Body thinks, oh, something bad is coming. The body thinks something is bad is coming. I need to do some response now, right? Then they start releasing histamines, right? These histamines are released continuously, and that will give you so-called allergic response. Now, when you get allergy, they say that go and take cetirizine, antihistamines, right? It's the same phenomenon. So somehow to reduce the histamine content, and that's how we can create allergy. See, antihistamines are very important because especially they give it when you are running nose and so on. If you get a bubbles after eating, they will act so fast. Within half an hour or 1 hour, you get less response, right? I always carry histamine in my back pocket because I'm allergic to certain things. I'm allergic to dust. So This is exactly the mechanism, right? There is something called antibody. They want to protect you. They're all protein. They want to protect you by some unwanted things. Sometimes they go crazy. They're drunk, maybe, right? I don't know. They think food molecules also has allergen or antigen. So when they interact, they create histamines, and that may be problematic. Now, plant proteins can be allergens, act as allergen. Not for everybody. It's a very individual thing. Not for everybody. Not universal. Okay. And plant proteins can also carry some other things. They can also act as allergens. They can create immune response, can damage your body. Am I clear? So this is about allergenicity in 2 minutes. But your immunology professor will talk about 2 months about all these things. Innate immunity, active immunity, passive immunity, immunoglobulin E, and there are many things are involved. I'm not an immunology professor for now, for today, okay? So how do we evaluate the allergens? Proteins can be allergens. For example, soy proteins contains hell lot of allergens. Okay, you can use STS page to evaluate. You can use size exclusion chromatography to evaluate them. You can use mass spectrometry to evaluate them. Okay, some proteins are allergens. And you can also use allergenicity evaluation by using immunological techniques. Have you heard about ELISA? Enzyme linked immunosorbent assay. It's purely based on antibody antigen interaction, right? So your protein might act as an antigen here, and there will be an antibody specific to that antigen. Reaction takes place, color change happens, they measure antigens and antibodies. Does that make sense? And I don't explain this one. So people often use ELISA assays and protein characterization to find out how much allergens you have in the food samples. Am I clear? Now, just to show you, I think there is one slide got deleted. I see one slide got deleted, but that's okay. I'll explain you. Now, please note that you have soybean, okay? And soybean has proteins inside. I told you last time, right? And proteins are located inside soybean, right? Allergens are also located inside soybean. And we don't eat soybean directly, do we? You get really a problem. It's cooked. So allergens can be inactivated. I'll talk about that later. But how the plant-based world is acting today, how they're producing, they don't use soybean directly. They get soy protein isolate, right? They get soy protein concentrate. How do we get that one? Go back to my second lecture. We do dry fractionation and we do wet extraction, right? During extraction, what happens? Let's say dry fractionation, you have soybean flour, you get fine powder and coarse powder. Now the anti-nutrients or allergens are actually fractionating, right? And the same thing happened with the wet extraction. There might be a possibility that you're extracting or separating, you're also concentrating unwanted things in each fractions. So keep that in mind. It's not that when you do purification, everything is gone. You may be up concentrating unwanted things also. Okay, I'll explain that in detail when it comes to anti-nutritional factor. Just to let you know that you may reduce the content of allergens, but it's not that you're completely removing them, even after doing the extraction and purification. So how do we remove the I'm sorry, I'm mixing up allergens and anti-nutrients. I'm very sorry about it because I'm also hungry now. So allergens, how do we inactivate them? A lot of methods. The easiest one is thermal denaturation. For example, you heat, proteins will be denatured, and then they might not interacting with the antibodies. Okay, lot of methods are being done. You can do the polyphenol cross-linking. For example, the antibody has a specific binding sites. Now imagine that is binding to this amino acids. Now you convert this amino acid into something else. Now the antibody cannot interact with them. So you can reduce the allergenicity. I don't explain all these things, out of the scope for me today. Just to let you know that there are a lot of different strategies to avoid allergenicity. So food has to be processed I was talking about a dilemma between processing and non-processing. If you don't process the food, you have problem with allergenicity. You can't eat everything raw. You can't eat everything gently processed. Sometimes we need to do heavy processing to kill some unwanted things. For example, allergens. Am I clear? You can also use enzyme hydrolysis because this is an allergen protein. Imagine you do the enzymatic hydrolysis, you get small peptides. Maybe they are not interacting with the antibodies. So you can reduce the allergenicity.
So, if a person is allergic to protein,
Yeah.
can he or she intake plant protein food or any kind of protein foods?
As I was explaining, it's a specific proteins are responsible for allergenicity. So if the person is allergenic to beta conglycinin and that person is allergic to plant protein, soy proteins now for example, it's very specific to specific proteins and specific human being. Allergenicity cannot be generalized. I am not allergic to soy protein, I can tell you directly. I can eat soy protein without any problem. Not my friend, he is allergic to soy protein. Now I can't generalize, say, oh, shellfish is allergic for you, now you eat more plant proteins. Maybe that person is also allergic to plant protein, I have no idea. So it has to be tested person by person wise. Did I answer your question? There's a big list of allergens here. I really recommend go and read this paper. They have made a big free for 4 big tables to showcase how many allergens you have in plant-based. I don't say that don't eat allergens, don't eat plant-based. That's a very bad idea because as I said, he might be okay with cereals, but he might not be. It's very individual. Okay. And he might be allergic to if he eats soy flour bread, but he might not be allergic if the same bread is prepared by soy protein isolate. Am I clear? There's a lot of processing involved that can change the allergenicity. Okay. I have some examples. I don't cover everything just to show you that we can use immuno-based, ELISA-based method to evaluate allergenicity. When you have time, let's just go through it. Now I'll move on to the next interesting topic called anti-nutritional factor. I'm pretty sure I'll cover it by 45 and you can go and have your lunch. Is it okay? Just bear with me until I cover anti-nutritional factor. Anti-nutritional factor, as the word says, they are the components, they compromise the nutritional value. Okay? They are the components, they compromise the nutritional value of protein in this particular case. Okay? Protein has to be digested, but sometimes they become not digested because you have an anti-nutrition factor associated with them. There's a lot of anti-nutritional factor. Okay, skip this question. I forgot about this question. We all know that we can't use cereals directly. Like let's say soybean. I already asked this question. We need to cook it, right? We can't eat. Some people can't eat every nuts. They need to be roasted, right? Because of cooking, roasting, germination, we can actually degrade this anti-nutritional factor. Then you can consume them the food, right? There's a lot of anti-nutritional factor associated with proteins. They are listed here. Lectins, phenolics, phytic acid, protease inhibitors, saponins, tannins, and so on. And I'm going to cover some of them very briefly. Is it okay? Did you understand what is anti-nutritional factor? They are the one compromise the digestibility or the nutritional value of proteins. Okay? Just to give one example, soybean seeds, it contains lot of anti-nutritional factor. Can you see? Soybean seed contains lot of anti-nutritional factor. To eat soybean, either you have to cook it or you have to process it. By processing and cooking, you can degrade most of them. Now, how do we get soybean? I just was mentioning this slide. We do it by dry fractionation or wet fractionation. During fractionation, the anti-nutrition factor is also fractionated. Okay, Sometimes they come along with protein, sometimes they go along with the carbohydrate. So, if you're doing wet extraction, last time I was talking about wet extraction, you took the flour, you add salt containing water, you add sodium hydroxide containing water, right? Then you get lot of fractions. Okay, we say that this fraction containing 70% protein, but you're also fractionating anti-nutrient in the fractions. So don't be fooled that fractionation will always help you to get only proteins. You're also fractionating anti-nutrients. So now the protein contains 70% protein, maybe it contains 10% anti-nutrients. So whenever you do the extraction studies, be careful where you are fractionating the anti-nutrients also. Am I clear? So based on the type of effect they pose on your body, or especially on the proteins, you have two classes of anti-nutrients, protease inhibitors, and then we have other things called tannins, phytic acid, and lectins. I'll briefly cover some of these anti-nutrients. Please note that there is a lot of anti-nutrition factors for minerals, vitamins, and so on, but I'm mainly focusing on the proteins. Okay. The first one is trypsin inhibitor. In the stomach, trypsin is an important enzyme, right? Not in stomach, in the small intestine. That is involved in the digestion of proteins. Do you agree? In the small intestine, we have trypsin. It's an important enzyme involved in the digestion of proteins. Now, plants have some kind of trypsin inhibitors. They're also proteins, basically. There are two classes of trypsin inhibitors in soy, well characterized. They call it as STI, Kunitz type soybean trypsin inhibitor, and Bowman-Birk inhibitor. Basically, these are the scientists, they have identified them. Two types of trypsin inhibitors are there. What they do is, they have trypsin, and this trypsin inhibitor will go and bind to the trypsin, create a very strong complex. Now, what is the job of trypsin? To digest the protein. So it wanted to go and attack the protein and digest it. Now it can't do that job. Why? A trypsin inhibitor is binding and interacting it with this, so it cannot go and attack the protein. So it inhibits the digestion. So that's a problem. So this trypsin inhibitor can come along with the purified fraction of your protein. This trypsin inhibitor can already present in cooked soybean. So that's the reason why people say plant proteins are generally less digestible, because they do have some trypsin inhibitors. Am I clear? The job of trypsin is to digest protein, but trypsin is now inhibited by a trypsin inhibitor, which is another protein, which is less digestible, plus it will not allow trypsin to attack the protein. Am I clear? Any question? And the trypsin inhibitor content, there's a table here. We have about 43 to 84 trypsin inhibitor units per milligram of sample here. And you can see that with compared to soybean, some other proteins have less trypsin inhibitor. Can you see? When you say soybean is 100%, you can use black gram. Pea contains very small amount of trypsin inhibitor compared to soybean. So that's the reason why we need to use a variety of different foods. Soy may not be sustainable, plus it also have a lot of anti-nutrients, especially inhibitors. Either you remove it, if you don't remove it, you have a problem. Okay. I don't cover this trypsin inhibitor assay. It's basically there's a lot of methods to analyze trypsin inhibitor activity, enzymatic assays. It's very easy. I will leave the slides for you with the citation. You can look into how to do this experiment. Okay. Other important anti-nutrient present in the plant is phytic acid. The phytic acid is basically a chemical that binds to metal ions. Okay, it chelates, let's say, iron. It can chelate zinc, right? Iron, zinc, and everything is important for our body. Do you agree? Now, if you have a phytic acid, it will bind to the iron that is important for our body. Now, the iron is not bioavailable, so you might get iron deficiency. And it is one of the problems of phytic acid, right? And phytic acid can also bind to the protein and make insoluble protein complexes. Now the protein becomes less digestible. Am I clear? Phytic acid can do two jobs. One, it can bind to the metal ions and makes metal ions less bioavailable. And phytic acid can also interact with proteins and makes protein less digestible because actually they compromise the protein solubility. So there are a lot of methods to assess phytic acid. In our laboratory, we use three methods, and we are developing the chromatographic method also. I'll briefly explain you how we measure the phytic acid content. Only one I'll explain. The phytic acid sample we take is a sample containing phytic acid. Okay, we add an enzyme called phytase. The phytase will hydrolyze phytic acid into myoinositol. Then we add another enzyme called alkaline phosphatase. Sorry, phytic acid will hydrolyze into myoinositol phosphate. Then we add another enzyme called alkaline phosphatase, and that will convert phytic acid into phosphorus inositol. And that phosphorus is measured indirectly using molybdophosphoric acid, and basically you get a blue color, right? It's an indirect estimation. We convert phytic acid into phosphate, and the phosphate is measured in the colorimetric assay, and the blue color intensity is directly proportional to amount of phytic acid. This is the very common assay we use in our laboratory today. Okay. We also have this is not important. We also have another assay is called ferric ion assay. In this case, we take the sample containing phytic acid. We know that phytic acid binds to iron. Okay. So we add an iron excess in this one. If there is a more and more phytic acid is present, it will bind to more and more iron 3 we have added. Okay. And the remaining iron 3, which is not bound to phytic acid, is measured using this bipyridine, and it gives I think a colored complex called yellow color, if I'm not wrong. This is yellow colored, and that can be indirectly measured. I'm not going in detail, it's not important. I just want to tell you that there are different methods available to measure phytic acid. One is a megazyme kit. Yeah. What is the color? Pink. Okay. I don't recall pink. Have you done this one? It's a pink color. So basically the idea is nothing but you take a phytic acid containing solution and add excess ferric chloride. The ferric chloride will react with phytic acid. The remaining ferric ion will be reacting with bipyridine, and that will get you the pink color complex that can be indirectly measured. It has its own advantages and disadvantages that I will skip it. So you can use these two methods to quantify phytic acid. And then we have another anti-nutrient called lectins. Lectins are nothing but carbohydrate binding proteins. They love carbohydrates. Okay, lectins loves carbohydrates. What they do here is that in the body we have proteins, and they're connected to sugar. Okay, in the body we have proteins, they're connected to sugars, and we call it as glycoproteins. Okay, and lectins can also like glycoproteins, and they start interacting it with the glycoproteins, and they can compromise the digestibility. Okay, glycoproteins have a huge role. I don't go in detail today. So they compromise digestibility, especially in the small intestine. Okay, that is brief explanation. Just remember that lectins are carbohydrate binding proteins. They can interact with the glycoproteins in the body and compromise the digestibility. And here is the table that shows how much lectins we have in plant-based. 2.5%, 3%, 1%, a lot of lectins are present. But most of these lectins are purified when you do the isolate preparation and concentrate preparation. So they are gone. But when you eat a whole seed, there is a lot of lectins. Am I clear? Then we have a glucosinolates. These are very characteristics to this Brassicaceae family. For example, mustard or rapeseed, and they're all containing glucosinolates. And the glucosinolates are actually good in some senses. In some cases, they are bad because there is an enzyme called myrosinase, and that can cleave glucosinolates and convert glucosinolates into thiocyanate compounds. They can cleave this moieties. And if thiocyanate or isothiocyanates are not good for the body, because there's a lot of theories, they say that they can interact with the hemoglobin and can avoid the oxygen carrying capacity of hemoglobin. There's a lot of problems. So they are considered as anti-nutrients. They can come along with the proteins. So here is the big table showing glucosinolates content in different in rapeseed, different glucose. There's a lot of 100 different type of glucosinolates. They are varying in the different concentration, especially in the rapeseed. So we can identify and quantify them. Then I'll come back to the tannins and phenolic compounds. They are also anti-nutrients. This one I've already explained in my 2 lessons before. You remember plant proteins are colored, okay? And because of the phenolic compound, polyphenols, okay? Some people call phenolics are very healthy, bioactive. A lot of people are doing antioxidant activity of phenolic compound, antioxidant activity of anthocyanins. The literature is kind of biased here. Some people call it as anti-nutrients. Some people call it as good bioactive. There we actually debate each other. I don't know which one is right here. On a protein context, okay, on a protein context, they are anti-nutrients. Exactly. Again, it's a debate thing. I read 100, there is a 2,000, 20,000 papers on phenolics and antioxidants. Everybody does. Total phenolic assay is the most common assay done by all the bachelor's, master's, and PhD students. TPC analysis. They love doing that. Easy. Total flavonoid assay. Very easy to do. We do everything. Oh, my extract is antioxidant. Wow, wow, wow. Super. Let's not go back to that story. It's nice to do, but it's some glitchy thing. Okay. I'm not going to all these things at the interest of time here. So phenolic compounds, what they do is, this is protein, right? I am eating this one. I need to digest it in my stomach, right? Now these proteins come along with the phenolic compounds, right? Now the phenolic compound I told in my previous lesson, I gave a lot of explanation. They attack to the protein. They can interact with the protein covalently and non-covalently. They can make a protein phenolic compound complex. Now this complex might not be digestible in our body because enzymes are very specific to peptide bonds, right? I taught you that one. Our proteases are very specific to peptide bonds. Now there is a peptide bond here, C double bond O NH, okay? Next to this peptide bond, I have a phenolic group attached, a very big phenolic compound attached here, this one, okay? Now my enzyme is coming. It wants to attack this peptide bond. What it will happen? It cannot access it because enzyme should go and sit on the substrate to cleave, right? Now we cannot access it because you have a big phenolic group attached like that one. Am I clear? Now the accessibility is a problem, so the digestion can be compromised. Not only that, these phenolic compounds directly sit on the enzymes because enzymes are also protein. So it will inactivate the enzymes. So you compromise the digestion. So 2 pathways. Number 1, enzymes cannot access the protein because of the bulkiness of the phenolic compound. The second one is phenolic compound directly interact with the enzymes, can inactivate enzymes, it can bind to its active sites, then enzymes of no use. So it will compromise the digestibility. Is it clear? So it is an anti-nutrient on a nutritional point of view, not a bioactivity point of view. That's what Asta was telling, right? For proteins, it's an anti-nutrient. Here is a table that shows different amount of anti-nutrient tannins in different foods. I don't read the numbers here. When you're free, just go and see, just to let you know that plant-based proteins can come up with a lot of phenolic compounds, okay? This is a summary table. It's also for the homework. This table summarizes different anti-nutrients compounds and how they act and so on. Now in 5 minutes, I'll talk about how do we eliminate anti-nutritional factors. I'll not go in detail. It's not the scope of today's lecture. You can do too many things to kill the anti-nutrients. You can do germination, you can do soaking, you can do heat treatment, you can do fermentation. Lot of things you can do to remove anti-nutritional factor. Because imagine, just to give one example, trypsin inhibitor this is trypsin inhibitor, okay? Trypsin inhibitor. It's a protein, do you agree? It's a protein. Now, if I add an enzyme, if it will cleave the trypsin inhibitor, now trypsin inhibitor is not active any longer. Am I clear? If you heat it, the food, if you heat it, trypsin inhibitor can be inactivated because it will lose the three-dimensional structure. Now it is no longer an active enzyme. Am I clear? So you can do heating. But every method cannot be used for everything, because if I heat it, you will also denature your protein. Okay? You will kill the trypsin inhibitor, that's right. But you will also denature the protein. So you need to find out the right method for right sample at the right time to get the right benefit. Am I clear? So there is no universal method to remove all the anti-nutritional factor. You need to try with so many things, and one of things will work. We're not talking about removal of anti-nutrient factor 100%. We're talking about reduction. That's enough for us. Okay. To a small level, anti-nutritional factor doesn't have any problem to your body. It's only they're present in a high quantity. They compromise the digestibility. Am I clear? So this is the last slide of today. Coming back to Asta's point, sometimes we call it as anti-nutrients, sometimes they are good. For example, phytic acid is an anti-nutrient for proteins and minerals, but it could also be healthy because minerals are sometimes problematic. Some minerals have to be chelated, then phytic acid will do that job. Phytic acid is a fantastic antioxidant. Anti-nutrients, antioxidants, they are different. But phytic acid is doing both the job, right? Polyphenols, and she says if you drink a glass of green tea, everybody says, oh, drink green tea is good for your health, right? I'm telling it's an anti-nutrient. It is a good antioxidant, it can do cardiovascular prevention effect, it has anti-diabetic, every property is associated with polyphenols. But for proteins, it's anti-nutrient. So what I wanted to tell you here is that don't take the term anti-nutritional factor so seriously on a generic word. It's anti-nutritional factor on a protein world. Okay? Their effect is depend on how much you eat and together with what you eat. Maybe it's a bad idea to drink green tea together with a particular protein. Maybe it's a bad idea to add milk to green tea and drink it. Maybe both are not bioavailable now. I don't know. I'm telling about the research we are doing in our laboratory, right? So it's more about how you're mixing do things, to what extent we are eating, and that determines their anti-nutritional value or bioactive value. We as a food scientist have to be very careful about using the terminologies. So that brings to the end of my lecture. I don't recall the number. It's more about allergenicity and anti-nutritional factor. So to sum up, what did we study today?
So first we talked about the how to evaluate the protein quality, because
Yeah.
when we are taking the alternative proteins, we need to focus on the protein quality as well.
Not
And
only
that
quantity, is quality matters.
quality.
Yeah.
And that can be determined in two ways.
Yes.
First is digestibility, and
Yeah.
in other terms we can say it bioavailability as well.
Yeah.
And about the amino acid composition analysis.
Absolutely.
It's
Yeah.
so we talked about how we can determine these both,
Yeah.
right? And bioavailability and digestibility can be performed in two ways,
Yeah.
using two models: in
Yes.
vivo and in vitro.
Yes.
In vitro, that we carry out the analysis in test tubes,
Yes.
right? And
Infogest
in
model.
infogest model.
Yes.
And they have two basically models, like static and dynamic. Dynamic, we have quite modern instruments which
Yeah.
make the stomach effect.
Yeah, they mimic the stomach effect. Yeah.
And
Yeah.
in static model we have infogest.
Yeah.
And in vitro we perform in two ways: PDCAS
In
and
vivo, we perform in two ways. Yeah.
PDCAS and
AAS.
DIAAS,
Yeah.
right?
Yeah.
So PDCAS is performed basically in mouse
and rats.
in rats,
Yeah.
right? And DIAS is performed using pigs model.
Yeah.
Right.
And we believe that DIAS is better than PDCAS.
Right.
Yeah. Yeah.
Then we talked about the amino acid composition analysis, because essential amino acids are very much important. So we have to analyze what is the composition of that protein in terms of amino acid.
Yeah.
So we utilize, first we have to break down that protein into smaller components, that is amino acid. Then we can use GC, FID and UHLC, right, with the FI detector.
Yes, fluorescence detector.
Fluorescence detector.
Yeah.
And then we talked about the allergenicity
Yes.
and the anti-nutritional factor. In the allergenicity, we know that there are typically all over the world 8 major allergens are present.
Yeah.
And our body recognizes some protein as antigen.
Yes.
Means our antibodies think that they are bad for our body.
Yeah.
So our innate immune system activate, and it gives symptoms in the form of rashes, respiratory effect, or indigestion.
Yes.
So and after that we studied about how to eliminate those allergen or how to detect those allergen. We have ELISA method and skin allergy detection test also. And after that we talked about the this anti-nutritional factor. The major anti-nutritional factors that we talked about was directly they are binding the protein. They are making the protein unavailable for the human body.
Exactly.
So major were lectins, trypsin inhibitor, and
phytic
acid,
tannins. Yeah.
So they do have the negative effect on the body, but they have few health benefits on the body. But still we have to remove so that we can get the maximum of the protein. So we talked. Just summarize the methods to eliminate those anti-nutritional factor as well. The physical and biological method. We can do it by soaking or heating. And in terms of biological treatment, we do it with germination or fermentation or other enzymatic treatment.
Absolutely. That is a fantastic summary, Asta, and claps for Asta. So well done. Thank you so much. And I don't want to summarize again. You've done my job. Thank you so much.